The overall goals of this method are to generate and identify in vitro cultured giant phagocytes derived from circulating human neutrophils for the investigation of their role in inflammatory and anti-inflammatory immune responses.
This method can be used to obtain and identify a new subpopulation of giant phagocytes, which develop from human neutrophils that are maintained in long-term culture conditions. This technique can be used to further investigate the significance and functions of giant phagocytes, as well as to answer key questions about their activation and plasticity. Demonstrating the procedure will be Eva Leder, a research assistant, and Oksana Rogovoy, postdoc, both from my laboratory.
Use a sterile scalp vein set to obtain at least 40 milliliters of venous blood from a healthy young volunteer. Then, gently mix the blood in a biosafety laminar flow hood. Dilute 10 to 12 milliliter aliquots of the whole blood sample to a final volume of 24 milliliters with pre-warmed ion-free PBS containing 2% heat-inactivated FCS. Then add 12 milliliters of polysucrose 1119 to the bottom of one 50 milliliter sterile polypropylene conical centrifuge tube per blood sample aliquot, followed by the careful layering of 12 milliliters of polysucrose 1077 on top of the polysucrose 1119 gradient solution. Carefully pipette 24 milliliters of the diluted blood onto individual tubes of the density gradient solutions and separate the cells by centrifugation.